Hi everyone, today I will share insights from our recent lab on enzymatic activity, specifically focusing on lactase and its potential implications for addressing cancer, a global health challenge. Cancer remains a leading cause of mortality globally, with an estimated 20 million new cancer cases and 9.7 million deaths in the year of 2022 from the report of World Health Organization. The complexity of cancer, its causes, and its progression highlights the urgent need for innovative treatments. Our lab's work on enzymatic activity, particularly through studying the lactase, offers a window into potential therapeutic strategies. And I will subsequently offer additional context derived from the lectures we learned. So we have learned from these lectures that enzymes are a specific type of catalytic protein that accelerates reactions without being consumed in the process. We've seen examples like sucrase, an enzyme that catalyzes the hydrolysis of sucrose. The underlying mechanism involves enzymes accelerating particular reactions by reducing the activation energy barrier, without altering the net change in free energy between the reactant and products. During the lectures, it was also emphasized that enzymes exhibit specificity for their substrates. Initially, the substance an enzyme acts upon is referred to as its substrate. The enzyme attaches to its substrate, forming an enzyme-substrate complex, during which the enzyme's activity transforms the substrate into the product. The reaction each enzyme catalyzes is highly specific, a phenomenon known as enzyme substrate specificity. The part of the enzyme where the substrate attaches is called the active site. Moreover, the concept of induced fit was introduced, where the substrate's binding causes the active site's chemical groups to align in a way that enhances their catalytic effectiveness. Furthermore, it was discussed that in an enzymatic reaction, the substrate connects with the enzyme's active site. Enzymes work incredibly quickly and are left unchanged after reactions. Even minimal amounts of an enzyme can significantly impact metabolism due to their reuse in catalytic cycles. It was also conveyed that the pace of enzyme-catalyzed reactions can be increased by elevating the concentration of the substrate. When every enzyme molecule's active site is occupied, the enzyme reaches a state of saturation. Once saturated, the only way to accelerate the reaction rate is by introducing more enzymes. Environmental conditions also play a role in enzyme performance, with an enzyme's activity being influenced by general environmental factors like temperature and pH, along with specific chemicals that affect the enzyme. Enzymes are crucial, and regulating their activity is key to managing the metabolism. Based on the previous introduction of the enzyme and lactase, I will now introduce the lab experiments. The purpose of the experiment I conducted was to evaluate the enzymatic activity of lactase at 37 degrees Celsius, utilizing a spectral photometer for measurement. As we introduced before, enzymes are catalytic proteins that accelerate chemical reactions without being depleted in the process. In this study, I focused on the function of lactase, a member of the beta-glucosidase enzyme family that plays a crucial role in the digestion of lactose in humans. Lactase facilitates the breakdown of lactose by splitting the beta-glucosidic bond, resulting in the production of two monosaccharides, galactose and glucose. We employed a spectral photometer, setting it to a 450 nanometer absorbance wavelength to measure light absorbance, which reflects the concentration of substrates and products. For the purpose of this experiment, OMPG, a compound that mimics lactose structure and is detectable by the spectral photometer, was used as the substrate. In this experiment, lactase was obtained by extracting it from commercially available lactase tablets. The tablets were crushed into a powder, dissolved in a solution, and then centrifuged. with the supernatant collected to create a lactase solution of a relatively high concentration. The hydrolysis of the samples occurred at 37 degrees Celsius and was halted at 30 seconds, 60 seconds, 90 seconds, and 120 seconds intervals using sodium carbonate. Light absorbance readings of the samples were taken to monitor the hydrolysis process at these various times. By plotting the data on a graph, the catalysis rate was deduced from the slope of the curve generated from the absorbance measurements. This experiment effectively demonstrated the characteristic of enzymes and the methods to regulate their activity, offering valuable insights into potential cancer treatments. Cancer cells, known for their rapid growth and division, possesses abnormal cell membrane that allows them to multiply uncontrollably. This experiment effectively demonstrated the characteristic of enzymes and the methods to regulate their activity, offering valuable insights into potential cancer treatments. Cancer cells, known for their rapid growth and division, possesses abnormal cell membranes that allow them to multiply uncontrollably, leading to lethal tumors. The key to their seemingly endless proliferation lies in the expression of the telomerase gene, which extends their telomerase, granting them a form of immortality. In contrast, telomerase genes are inactive in most somatic cells, with the exception of certain stem-like cells and T cells. Drawing on this knowledge, I'm positioned to suggest a potential therapeutic strategy. For the real-world connection part, I would like to draw parallels between our lab work and the cancer treatment. The principles of enzymatic inhibition provide a compelling framework for the cancer treatment. As we all know, cancer cells is characterized by unchecked division and immortality via active telomerase. This poses a significant challenge for treatment. In our lab, we have observed how targeting enzymatic reactions with compounds like sodium carbonate can effectively halt biochemical processes. Similarly, conceptualizing strategies to inhibit telomerase present a novel avenues for cancer therapy. Telomerase inhibition, however, comes with a set of critical challenges. Firstly, it is a long-term strategy requiring sustained intervention and monitoring over multiple cell generations. This slow attrition strategy means that eradicating cancer cells is not an immediate process, but rather occurs gradually as telomerase shorten and cell proliferation is impeded. Secondly, there is a notable risk of collateral damage to healthy cells, especially those with stem-like properties, and T cells is crucial for the body's immune response. These cells rely on telomeres for their regenerative capabilities and to maintain functionality over time. The key lies in developing treatments that selectively target cancer cells while sparing healthy ones. Precision medicine approaches, such as creating targeted enzyme inhibitors for telomerase that either compete for the active site or alter the enzyme's shape, hold promise in achieving this goal. By doing so, we aim to minimize adverse effects and ensure the safety and well-being of patients undergoing this treatment. Enzymes offer significant potential in advancing cancer therapies by providing targeted interventions from this lab. Fine-tuning enzyme activity allows for more nuanced approaches to treatment. This emphasizes the need for continued research and innovation in this field. And this experiment underscores the importance of exploring enzymatic mechanism in medical science and highlights the pivotal role enzymes can play in enhancing cancer therapies. To conclude, I would like to express my deepest gratitude to Professor Li and the lab instructors for their invaluable guidance, to the Kunshan University for providing a nurturing academic environment, to my classmates for their collaboration and encouragement, and to my family for their unwavering support. This journey has been profoundly enriching, offering both scientific insights and personal growth. Thank you so